Then I'll start. Good day, Боревдзе, здравствуйте. I'll just switch off my video and I'll leave only, I think, the screen. I was asked to talk about our works on origin of life, although these are not the only works that we do. And therefore, I'll give a talk which is separated in the following sections. First, I'll give some introduction in what we know about very first organisms, because in principle there is some knowledge about them, and I'll speak about that, but very briefly, because all the stuff I'll discuss is published. Then I'll talk about paradoxes which are related to origin of life, and there are many of them actually. I'll speak about 3 of them, and that's what we do not understand yet. There is no consensus. And at the very end, I'll consider one of possibilities to solve these paradoxes by combining the knowledge about living organisms with some rather unusual set of data which was never involved in this origin of life problem. And I'll proceed because I have a lot of slides and I'm limited in time, therefore I'll speak pretty fast, but I think it's not a problem in this case. Now, first, what biology tells us about origin of life and the first life forms? Basically, I hope that everyone knows in this audience that the major biopolymers are DNA, RNA, and proteins. And it was discussed starting from the very beginning, which of them came first. And all these polymers are long, they can form particular structures, they have different properties. But if you compare what these polymers can do, then it comes out that DNA can store information, but DNA molecules can hardly catalyze chemical reactions. Proteins are great catalysts, but they cannot store information. And RNA molecules, they can do both. Because they can catalyze chemical reactions, they can form kind of coiled structures with catalytic activity, and also they can store information because RNA viruses, for example, the pandemic virus of today, they store all their genome as RNA molecules. They don't have DNA at all. And therefore, it was suggested that very first organisms perhaps were kind of RNA molecules. For the first time, it was suggested in 1957 by Andrei Belozersky in Moscow, and I'm from institute which is carrying his name. And later, some 11 years ago, American scientists came to the same idea. Now, what we can say about RNA work, it appears that we cannot say much. And generally, all this subject of origin of life is rather special because everything happened many billions years ago, and this meeting is devoted to Timofeyev Rysovsky, and it's well known that when he was asked what he thinks about origin of life, he usually answered that he was very small at that time and he don't remember. And that's true. We do not remember what happened 4 billion years ago, but our molecules do, because they are direct descendants of the very, very first biological molecules. And specifically, which was done during last decade and which passed somehow unnoticed, was the reconstruction of evolution of ribosome, a cellular complex, very complex actually, which is making proteins, reading the information which is stored in the genes via RNA molecules. And it's a huge machine. I have no time to go into its structure and functioning, but the interesting thing is that in 3 different labs, very good labs using different methods, the evolution of the ribosome was reconstructed. So we understand pretty well how it happened, strangely enough, although it happened again, I repeat, 4 billion years ago. And what comes out from this reconstruction, these are very interesting papers, I just can suggest to read them. But what comes from this reconstruction is the following. The ribosome, it is built of two large subunits made of RNA mixed with proteins, and the evolution of these subunits was reconstructed, resurrected. They went back and understood how the very first protein synthesizing RNA molecules were working. And at the very beginning, there were two very small RNA molecules, 55 nucleotides each, interacting with each other and connecting to amino acids together. And the reconstruction shows that, and these two small RNA molecules, they made a large ribosomal subunit at the very end. Now, as you see, a ribosome is a very complex, large system. And there is a small ribosomal subunit, which is interacting with RNA molecule, which is coding the information. It's decoding center. And decoding center is on the small RNA subunit. And all this reconstruction shows that these subunits met each other, so that proteins could be synthesized using the information from RNA molecule somewhere in the middle of the way. So for many millions of years, this large ribosomal subunit was evolving separately, shown here, and it means that it was connecting random amino acids with each other, and it was non-coded protein synthesis, and there is little doubt that that's so. So we know about RNA world, it was there, and this machine, which apparently was key for functioning of this world, could be reconstructed. And the RNA world started around a machine, in a sense, which was producing non-coded polymers, which did not carry any information. The machine was just working and making proteins from random amino acids. And if you speak about random amino acids, then these amino acids were synthesized somehow by nature, and in this case, most of such abiotically synthesized amino acids are very simple, and it's glycine, a very simple amino acid without any side chain, and it cannot catalyze any reactions. So ribosomes were producing mostly polyglycine, which is a kind of polypeptide, but it cannot catalyze anything because it doesn't have side chain with chemical groups which can catalyze chemical reactions. And the question is why it happened. There is little doubt that it started in this way, but the question is why it started this way. Another story is the reconstruction of the habitats of the first cells, and it's our work, which was published in PNAS some years ago, and it was done in collaboration with geochemist Andrei Bычkov of Moscow State University, and with genomic experts Michael Galperin and Eugene Kunin. Eugene will speak, I think, in this audience today, and also Daria Dibro, my PhD student, was involved. And what was beyond that? Eugene Kunin, some 20 years ago, by comparing the very first genomes of different organisms, have found out that there are about 60 protein coding genes, which are present in all living organisms. And therefore, since this set exists, we can say that all living organisms, they had a common ancestry, and this ancestor is called LUCA, last universal cellular ancestor. And because we have these 60 genes, we can say something about these LUCA. And if you look what these universal genes are doing, and they are coding for proteins, 40 of them, 2/3, are coding for proteins that are involved in protein synthesis. And other 20 are coding for something else. Some of them are coding for membrane proteins. Maria Kozlova will speak about that today briefly. But the point from this work is that protein synthesis was the core of the very first cells, and the cells emerged around protein synthesis. And this protein synthesis, as I've told you at the very beginning, was non-coded, just random polypeptides. And altogether, it's interesting and it's strange. What we're interested in the case of this particular work was the nature of inorganic ions that were interacting with the first cells. The strange thing is that inside our cells, the chemistry is completely different from marine water, from fresh water. We have much more potassium than sodium inside our cells. We have a lot of transition metals inside of our cells. And actually, if you compare it with seawater, it's principally different because in seawater you have much more sodium than potassium and so on. And therefore, what we have done, the very first cells, they could not have very tight membranes. Again, today it would be spoken about that. And therefore, they should be in equilibrium with small ions around them, because modern organisms, they have a lot of pumps which are always pumping ions back and forth, and these pumps are rather late invention. And therefore, what we have done, we have taken this list of 60 proteins present in all organisms, and these are the oldest proteins that we know. You cannot read this table, but it's it's not very bad. And we checked which inorganic ions are interacting with these proteins, on which inorganic ions their function depends, which inorganic ions are involved as structural elements in these proteins. And to our surprise, it came out that several of these proteins, mostly again involved with protein synthesis, depend strictly on potassium. And none of these proteins depends on sodium. And that's the reason actually why our cells are always pumping sodium out, potassium in, because otherwise protein synthesis doesn't work. But it also means that very first cells started under some unusual conditions where there was more potassium than sodium. And in collaboration with Professor Bychkov, we checked geochemistry where you can find such solutions. It came out that the only known solution is the condensate of geothermal vapor. Strangely now, geothermal vapor contains more potassium than sodium. There are some reasons for that, and more or less we're the first to notice that it's a very important feature. But you have geothermal systems, they are producing hot water and vapor, and chemistry is different, and vapor contains more potassium than sodium always, but also it contains organic molecules, carbon dioxide, hydrogen sulfide, ammonia, very important compounds if you are speaking about some very early evolution. And the places where this vapor is coming out are called geothermal fields. And we speculated that on such fields, the very first cells were living, perhaps in some cold pools of geothermal vapor. It condenses, and then the chemistry of these pools is also potassium rich. And interesting feature of such geothermal systems is that they are heated from bottom. And therefore you have different set of temperatures on such fields, and altogether it is independent of climate. You can have very cold climate, but it will be hot on a geothermal field, and in principle you can have any kind of temperature there because there is some kind of temperature gradient around geothermal vent. Therefore the Earth could be covered by ice completely, but around volcanic system, around geothermal fields, it should be pretty hot. Now I'm going to these paradoxes in the origin of life. And the first paradox that I will cover is the faint young sun paradox. And what's the problem? The problem is that at the very beginning, our sun was faint and weak and produced much less heat than today. And there is little doubt that it was so. And therefore, if the atmosphere of the same density as today, then the temperature on Earth was on average minus 25 degrees. And if the atmosphere was even thinner, then it should have been lower. And this was realized some in 60s and 70s, and scientists started to think. But scientists assume that you need liquid water. And you don't have liquid water at this temperature, and therefore they were thinking what to do. And the currently most popular solution, not the only one, but the most popular solution is that there was a very dense atmosphere of carbon dioxide from the very beginning, and there was a lot of water vapor, and there was a greenhouse, and because of this uh, uh, very dense atmosphere, the oceans were liquid. But I repeat, it's not the only solution, but it's the most popular. I present it. The problem with this solution is that there is not a single evidence that the oceans were there from the very beginning. And another problem is that we don't have any geological record uh, earlier than 4 billion years ago, because there are no rocks which are older. And the only thing that we have are small zircon grains, which are very stable, and once formed, they then cannot melt. And these zircon grains are studied by scientists with a great effort, because it's the only remnants older than 4 billion years. And these zircon grains, they report that they have formed under conditions of quasi-continental rocks, of granitic rocks. And they do not report on any conditions similar to sea conditions. Anyhow, another paradox, which again is in the room, is the water paradox. The water paradox is very simple. If we take biological polymers, any polymers, DNA, RNA, proteins, and you check the reaction which leads to their formation, in all the cases, it's a condensation. It's condensation, and it's a reaction during which water molecules are released. And it means two things. First thing is that all these polymers are not stable in water. Indeed, they are. They hydrolyze with time. And another point is that they hydrolyze, as shown here. And another point is that to produce these polymers, you should do something to take water away. Nowadays, enzymes are doing that. And these are very complex reactions. And in the absence of enzymes, this reaction could not run in water, because water will hydrolyze these polymers and not favor their biosynthesis. And there was spontaneous biosynthesis of proteins or RNA or DNA, whatever. It could happen only under conditions where the water was taken away. And therefore, chemists working in this field, they don't like water. So it's a real, very hard paradox. And how it could be solved. And chemists working in this field, they are investing a lot of efforts in that. And their current most popular idea is that the very first polymers were formed not in water, but in another liquid. And as this liquid, such liquid, they suggest formamide. It's a very small molecule, which is shown here. And it's a kind of a free-living peptide bond, because chemistry of formamide is exactly similar to the chemistry of connection between two amino acids in a protein. It's a strange similarity, and we'll come to that. And then if you take formamide instead of water, and then if you heat formamide, you get amino acids, different amino acids. You can illuminate formamide by UV light, you can use different catalysts, and you get nucleotides, you get amino acids, you get everything. Just you should replace water by formamide. And therefore, people working in this field, they coined the term of Formamide World. And you can just take a solution of formamide without any catalyst, illuminate by UV light, and you get these very complex molecules, which are called nitrogen bases, and which make the base of DNA and RNA. And there are different research groups. Some are starting from formamide, others are using cyanide as a starting compound. But all these compounds, simple compounds around nitrogen, are connected with each other by hydrolytic reactions. You add water to a cyanide and you get formamide, and the other way around. So it's one large piece of chemistry. And this is one of recent examples where, starting from a cyanide, collaborators of John Sutherland in United Kingdom, they produced nucleotides, activated nucleotides, which can polymerize on their own. and amino acids and sugars and whatever you want. So the chemistry is understood. It's a very nice chemistry. But this chemistry implies that these reactions proceeded in water. And what I have on this slide are also catalysts which are needed for these reactions. And you need phosphorus compounds, you have borate, rather strange set of different chemical compounds, but they are present in geothermal vapor, by the way. It's rich not only in potassium, but also in borate, in molybdate, and several other things which are important for this kind of chemistry. How formamide could accumulate? The trick is that formamide boils at 200 degrees of Celsius. It means that if you evaporate some solution, some mixture of water and formamide, then water evaporates and formamide stays there. And that's the base on which different scenarios are suggested. And one of the recent scenarios, but they are very similar to each other, is that there was a meteorite that hit Earth, and then it was a meteoritic crater, all the water evaporates, and the crater was cooling down, and at a certain moment some formamide amounts gathered there, and water still could not come into play. And then the chemistry starts, and the molecule started to form spontaneously. And later, when some kind of life was there, the water also came, and they adapted to water. And there are at least 3 such scenarios suggested by different groups, and altogether it sounds very reasonable from my opinion. The third paradox that I would like to mention is the zinc paradox, and it's a kind of our own problem, because when we checked in the same list of oldest proteins what kind of transition metals they prefer to have, it came out that many of them prefer to have zinc either as catalyst or as a structural element. And none of these proteins needs iron, and that's the problem, because in nature generally you have 1,000 times more iron than zinc. Zinc is not very rare, but it's a rare element, and strangely very oldest proteins are using zinc. And there is a huge machinery in our cells which is scavenging zinc from environment, concentrating it inside the cell. It's a separate field of biochemistry. But at the very beginning, you should have 1,000 times more iron, and why iron is not there, why the zinc is there, is completely unclear. Again, the geothermal vapor is nice because it concentrates zinc. The ratio between iron and zinc generally is 1,000. In the case of some water coming from the geothermal system, it's up to 500. And in the case of vapor, here separately chemistry of vapor and chemistry of geothermal water are shown. In the case of vapor, it can drop up to 2, but still you have more iron than zinc. And this zinc story, just many years ago, I have published personally as a kind of evidence for zinc world, that the very first organisms were living at high zinc. And actually, there was no good explanation how it could happen. It comes from biological data, but there is no geological model. And now I'm coming to the last part of my talk, and where I'll try to suggest some scheme which solves all these paradoxes. And it's about origin of life on dry earth, and perhaps someone can recognize that this is masses from the space, and it's a very dry place here, as you all know, because you are somewhere here now. I cannot directly see, but perhaps Aragats is somewhere there. Anyhow. Now, as I've told you, in these Formamide World scenarios, formamide was concentrated by evaporating water by heat. It's possible, of course, but it's not very nice because RNA molecules, they are, for example, very labile they are fragile at high temperatures, and it's unclear how they could survive. And therefore, there is another possibility to get the same enhancement in formamide and similar amide molecules. And in this case, we turned to one more meteoritic impact, but very special one, and it's the moon forming event, because mm, uh, generally it's accepted that moon formed as a result of a collision between Earth and a Mars-sized planet. As a result of this collision, a lot of Earth mass went into space and then somehow gathered to form the moon. And scientists believe in this event because in the case of major elements such as iron, oxygen, and so on, silicium, the isotopic composition of moon is exactly similar to the isotopic composition of Earth. So what kind of event was that? What was the size of the impact? What was the velocity? What was the angle? It's unclear. There are many ideas. But what is very well known is the geochemistry of this event, because there is 400 kilograms of lunar rocks brought by American astronauts, and there are some Soviet samples. And what these samples show is that moon rocks, on average, contain 100 times less zinc than Earth rocks. And also there are some difference in isotopic composition, and also moon is depleted in sodium and potassium as metals. And altogether, it's very well known, very well proven. And what is the explanation? And the explanation is that what zinc and potassium and sodium have in common is that they are pretty volatile metals. And zinc is, formally speaking, very volatile, the most volatile metal. It's a heavy metal, therefore it could not be lost as hydrogen to the space, but it could be lost in some scenario because of its volatility. This is the scheme with different chemical elements. There are refractory elements which evaporate at very high temperatures. There are volatile elements, and usually these are simple elements such as carbon or oxygen and so on. And there is this section of moderately volatile elements where the zinc is the most moderately volatile element. It evaporates at 600 degrees Kelvin, at pretty low temperature. And what planetologists think about all that? They think about, they need some scenario, and this is the last scenario, but the previous scenarios were also in this direction. The idea is that upon this moon forming impact, some planetary disk formed. This particular version is called synestia, which was very hot. And at the edge of this disk, the moon somehow condensed, and this disk gradually collapsed with time over thousands of years, but remaining hot inside. At a certain moment, it becomes smaller than the lunar orbit, and so the moon stayed back, let's say, but the volatile elements, which were still uncondensed at that moment, it means at temperature about 1200 degrees of Kelvin, they stayed inside of this toroidal disk. This is kind of animation which shows how moon is coming out of this disk, which in this recent version is called synestia. And it means that the zinc, potassium, and sodium of moon stayed inside this disk and was brought to Earth, which is interesting. What happened on the Earth? On the Earth, there is a set of scenarios how the Earth was cooling of the moon forming impact. And generally, this impact led to the loss of the atmosphere, according to most models, because of impact itself and very high temperatures. But then everything started to cool down. And the atmosphere was a silicate atmosphere because silicate was not lost. And according to these models, and it's kind of a review where the most planetologists working in this field are present. I show the list of authors, and I'm going along this scenario. It's not ours, it's just general thinking in this field. When the atmosphere cooled below 1600 degrees of Kelvin, the silica rain started, the silica starts to condense. And when the temperature goes below 1100 degrees of Kelvin, the silica start to become solid, and the first solid crust appeared on the Earth. And in this case, the Earth was covered by magma ocean. It was very hot, and then the cooling proceeded further via some volcanic systems existing in large amount on this very first crust. And then at even lower
You have only
temperature,
5 minutes.
yeah, at even lower temperature, the first moderately volatile elements started to precipitate on Earth. And what we see in this connection is that the zinc was precipitating last. And the very last level of this overcrust was made and very enriched in zinc, in metallic zinc, actually, because it has the lowest condensation temperature. And the Earth after the mega impact was dry and zinc coated. And then since the atmosphere was lost, the water could not stay in the liquid state. So it existed either as vapor or as ice. So we take seriously that the temperature were low. Yes, they were low, and I think we cannot do anything with that. But we have ice and we have a volcanic vapor. And this model was suggested in 1977 for a dry Earth by Lev Mukhin and Vasim Moroz. And in this case, we have a lot of geothermal systems. And around these geothermal systems, you have, of course, different temperatures and whatever you want. But because atmospheric pressure is very low, water is still evaporating. You don't have liquid water. And then around these geothermal systems, the life could start, because under these conditions of low atmospheric pressure, formamide did not evaporate, or formamide-like molecules, and generally molecules with high boiling temperatures were staying liquid, and this is glycerol, organic and fatty acids, and formamide, and there was a very special liquid medium, which was liquid, so it could provide conditions for chemical reactions, but there was no water there. And then under these conditions, the first replications in the absence of replicators, RNA-like replicators could start in the absence of water and evolve. Under these conditions, these nucleotides could polymerize, condense spontaneously. And so geothermal field, they could provide sources of organic compounds, metallic zinc as a reducing agent, it's very important, opportunities for concentration of reagents, and some liquids with a chemical composition similar to cytoplasm. But as the sun was warming up and carbon dioxide concentration increased, the temperature rised. And then some water started to come into play. There was some more water in these very first ponds and puddles. And this water was bad for the very first molecules because it was hydrolyzing them. And then these organisms start to find some solution and to protect them from water. And then I have started from saying that formamide is a free-living peptide bond. And actually, we can twist this statement and to say that peptide chain is a polymer of formamide groups. So by producing non-coded proteins, the very first RNA organisms were protecting themselves from water. They were just covering themselves with these proteins and surviving. And in these terms, the concentration of peptide groups in modern cells still is about 5 moles. We have 5 moles of formamide inside our cell, and the formamide world is still there. It's possibility. It's a possibility. Another part of the story is that this Earth, when I've told you that the Earth was zinc covered, it's exactly zinc covering that you use in your cars. The cars are covered by zinc from below to prevent the rusting, because as long as you have metallic zinc, iron cannot oxidize, because zinc is much more powerful reductant. And therefore, as long as these 10 in the power of 29 kilograms of lunar zinc were covering Earth, there was no free iron ions to interact with proteins. Therefore, proteins were binding only zinc ions because these were the only ions available for them, and we can explain this story with zinc-dependent old enzymes. And it proceeded pretty long until the temperature rose up, until
Sorry,
the
you have 1 minute on your
yeah, I'm through. Until the plate tectonics has begun, and then this zinc ring over crust of Earth underwent subduction, and it's somewhere now in the mantle. And this scenario that I suggest describes the transition from RNA replicators to the first cells as a series of adaptations to the gradually increasing water content at the surface of Earth, provides conditions for spontaneous biosynthesis of biomolecules in the Formamide world, explains the predominance of zinc ions in ancient enzymes that emerged in the zinc world, and describes the transition from the RNA world to the first cells. Uh, uh, I think that I'm through. Thank you for your attention.